Hello, I'm Harry Glorikian, and this is The Harry Glorikian Show, where we explore how technology is changing everything we know about healthcare. If you follow tech news, you know there have been a lot of stories over the last few months about AI chatbots like ChatGPT that can respond to your questions with incredibly convincing and well-written answers. These so-called large language models can tell you how to build a treehouse, how to bake a cake, or how to sleep better, and even write fake Bible verses about why it's a bad idea to put peanut butter sandwiches into a VCR. But notice the word large. The new generation of chatbots is effective in part because they have so much training data to work with. Behind the scenes, these models have sifted through hundreds of billions of pieces of data, basically the entire internet, including all of Wikipedia and thousands of published books, to see what words and phrases tend to occur together. So when you ask one of these large language models a question, it predicts the most likely answer based on a huge network of statistical associations. Now imagine that a chatbot came along that could learn how to generate convincing text responses by studying, say, only 18 sentences. Well, something like that is what my guest Raphael Townshend has accomplished for the world of structural biology. Townshend is the founder and CEO of Atomic AI, which is commercializing a discovery he made while earning his PhD at Stanford. He built a deep learning model to tackle the hard problem of predicting the 3D structures of RNA molecules. RNA has been in the news a lot lately too, in part because some of the vaccines that helped us beat back the coronavirus pandemic were made from messenger RNA, a form of the molecule that instructs cells how to build proteins, in that case, antibodies to the virus. But RNA has many other functions in the body, and if we knew how to modulate those functions, it could open up a whole new realm of medical treatments. But to do that, we need to know how each type of RNA is structured, so we can then design small molecule drugs that will attach to those structures. Just as a reminder, RNA is like DNA in that it's made up of a sequence of nucleotides. An RNA with a known nucleotide sequence can take many different possible shapes. And that's the hard problem. Townshend's deep learning model was designed to evaluate a proposed structure for an RNA molecule with a known nucleotide sequence and predict whether that proposed structure would turn out to be correct in nature. And it turned out it could do that with extremely high accuracy. So accurate that the discovery got published on the cover of Science magazine back in 2021. And what's remarkable is that Townshend's model required so little training data. It started off with just 18 examples of RNAs with known structures. So in a way, it was using the power of small data, together with a bit of physics, rather than the big data we usually talk about here on the show. Now, Atomic AI is building on that original model to create an engine for discovering new small molecule drugs that could interrupt any disease where RNA is a player, which is a lot of diseases. Townshend agreed to come on the show to tell us about it. Here's our full interview. Raphael, welcome to the show.
Pleasure to be here. Thanks for having me, Harry.
Yeah, so, well, first of all, super congratulations on your funding news. I mean, I know you guys just announced on January 25th that you guys raised 35 million in a Series A financing on top of the previous $7 million in seed funding, right? That's no small feat. For those
Thanks.
people that are listening, can you explain in broad terms what the company's mission is and what's so special about your technology approach?
Yeah. Um, at a high level, we really see atomic AI as sort of at the intersection of two of the largest breakthroughs of the last decade. I think everyone's heard of the RNA revolution with the mRNA vaccines, things like that. but also the AI sort of revolution with all these new sort of cutting edge AI technologies. And really, we see Atomic as operating at the intersection of these two, starting with my own work, which, for my PhD, essentially, which ended up actually featured on the cover of Science, to really create this new field of RNA drug discovery, right? With this cutting edge combination of RNA biology and AI technologies.
So, you know, I know I know you want to. We're going to come back to this a lot in sort of the way that I'm thinking about approaching this. But how does Atomic deliver on the vision you spelled out in the 2021 science paper?
Right. So there's been this sort of large amount of progress in AI specifically for structural biology, which is sort of the area of biology which has to do with the shapes of molecules. And really, one of the central mantras of structural biology is that structure determines function, right? The shapes of things determine what they do, which is really almost too obvious in some ways, right? Like the shape of a bike is important to the fact that it can transport you around. If
Right.
the wheels were in the wrong place, right, it wouldn't do a very good job of that. And so, very similarly, by understanding the shapes of the molecules, you can understand what they do and then also understand when things go wrong and then potentially intelligently design medicines to fix those things. And the progress on the AI side of things is that you can take any sort of molecule and really predict their shape overall, which is a process that typically can take months or years if doable at all. And you're bringing that down to seconds.
So, I, you know, I think the story of Atomic would make a lot of sense to listeners if we can talk a little bit about your personal journey, how the science paper got published, and how the company came together. I mean, along the way, we should talk about obviously the science of RNA, and all of that will help us set up like how you are, developing a drug discovery business. So first off, can you tell me the story of what you studied in grad school and how you got interested in machine learning to predict molecular structure? Because if I understand or have done my homework correctly, right, you were a PhD student in the laboratory of Stanford professor Ron Dror,
Yeah.
who is a computer scientist and not a biologist, if that's right.
Yeah, I would describe him sort of as a computational structural biologist, at least somewhat. I would say he has some, he's definitely has very strong sort of biology knowledge, but from a computational standpoint. Um,
Okay.
to give you a little bit of background as to, how we ended up here, there's a long version and a short version, so I'll try and stick with the short version. Um,
Okay.
I actually started my PhD in the artificial intelligence laboratory at Stanford, working on computer vision, actually, working on 3D vision, self driving cars, things like that. And really, there was sort of this realization that I came to that there was a lot of people that were trained very similarly to myself, working on the same problems. And I realized that this tool that is AI could potentially have impact broader in a much broader sense. And so
Mhm.
maybe a simple way of describing this, I kind of had this hammer AI, right? In some ways, and I'm going around looking for an area that is relatively neglected, right? In some sense, in terms of not many people working in that space, but that was very high impact. And so there's this rotation program at Stanford where you can rotate through different labs. And I essentially, rotated through Ron's lab, did no AI, and was just trying to understand the space as best as possible. And after that rotation, I realized, hey, this is actually seems like a very good fit. There's not many people working in structural biology plus AI. I feel like there's a few in the world at the time. But if you can realize that you can really unlock a lot of new potential in drug discovery. And, to make a long story short, then I had a 7-year PhD where I had to learn a lot of structural biology, essentially. But that actually ended up being quite successful at the end of the day, leading to this work predicting the structure of RNA, using these cutting edge AI methods. And overall, in computer science, it's actually fairly common to go straight from a PhD to a faculty position from there. And so, based on the success at first, that's the route I thought I was going to go. But I started writing these grants around using this technology, and two things became very clear. The first one was that there was a large commercial opportunity around, finding the shapes of these RNA molecules. And I realize I've only described that at a high level so far, so definitely happy to dig into that specifically. And the second was really that you needed to scale the amount of data that you were collecting to train these AI models to a level that would be relatively inaccessible in an academic context. And so through those sort of two key realizations, essentially Atomic was born about 2 years ago. And maybe I'll cut it off there for a second, but that's a lot of the genesis of Atomic, essentially.
Yeah, well, but it's interesting. I mean, you use the word rotation when you said it, and I was just thinking about, you know, ARAS, which, the the acronym you give it, right? But you
Yeah.
developed this model called ARES, ARES, if I remember correctly, for
Yes.
Atomic Rotational Equivalent Scorer. Um,
Yes.
and we come up with so many good acronyms in our world, but
Mm.
and it turns out that it really performed incredibly well at predicting whether a predicted RNA structure was true to sort of real world data. In other words, much better than previous method. I mean, was that a surprise when you did this the first
Yes.
time?
A short answer is yes. Um, you know, to dive a little bit more into that PhD, I actually started working on proteins and protein interactions. But the whole dream was really training machine learning methods on molecular structure, systems of atoms, or something like that.
Right.
Ron, my advisor, actually gave me a plaque at the end of my PhD that says "Atoms are everywhere." It's blurred out, but it's actually sitting behind me right now. But the fundamental idea is proteins, RNA, DNA, carbohydrates, et cetera. They're all made of atoms at the end of the day. And so if you
Mhm.
can design the right machine learning methods to work on systems of atoms, that should be fairly generalizable to new kinds of molecules. And so once we had our initial success on proteins, essentially we then sort of approached, this group at Stanford, Riju Das, that were experts in RNA structure, and said, hey, we've got these methods that work well in proteins. Let's try them on RNA. And that worked amazingly well almost immediately. And it was just very exciting at the time, and really sort of highlighted to me like how neglected in some ways this area was from a machine learning standpoint. Like if you could get that kind of amazing result so quickly, who knows what else is out there essentially to
Right.
achieve.
But let's take one step back, right? So, try delving into this. It when I was looking at it, right, if you just think about it objectively, it sort of seemed counterintuitive and unreasonable since most of the successful algorithms in the world of deep learning are trained on huge amounts of data. All right. And by the way, those are the kind of machine learning we usually talk about here on the show, right? Where the role of big data in medicine is sort of one of our big major themes. But the results you got in the science paper were based on just 18 examples of known RNA structure, right? At what point did you know that this was something that you could commercialize, apply to drug discovery, right? And the science paper only came out in like August in 2021. So it wasn't that long ago. So how do you think about those two things of small data, right, in some sense versus the large data? And how did you come to the epiphany that, wow, we can actually do something with this and make money?
Right. I mean, I guess there's two components to that. So the small data versus large data, right? Um, I think a lot of the key in the Ares example specifically was that we had to very intelligently design the machine learning algorithm to be data efficient, specifically, that it was able to sort of almost be aware of some of the laws of physics that drive, molecular structure. In particular, you mentioned the rotation side of things, right? if you rotate your entire molecule, right, it's still the same molecule at the end of the day. And more traditional machine learning algorithms essentially are unable to encode that directly. It kind of has to learn the thing separately in every different orientation. And so by building in this notion of rotational invariance, or really equivariance, which is a slightly related concept to all of that, we're able to be much more efficient and work off of a much smaller data set, right? Don't get me wrong, additional data can almost always be helpful, but in this case, getting even a single RNA structure can take months, right? And so you don't
Right.
really have the luxury of collecting very large amounts of it unless you're willing to spend a lot of time and money to really an absurd scale.
So I'm, hopefully I got this right, but if one of the big strengths of Aries was that it can predict whether a given molecule structure is accurate based on only a few examples of known structures. And it
Yeah.
doesn't matter what kinds of structures. In fact, if my understanding is correct, it doesn't need to know anything about what types of molecule it's modeling. It just needs to know where each atom is in a 3D space and what element each one represents. And well, first I should ask you, did I even get that right?
Yes, that is 100% correct. It's
Okay,
really
good.
3D space and what kind of atom, because at the end of the day, you could almost rederive everything else from that, right? If you knew where all the atoms were, you could back out which ones were bonded together, what, elements are, what bases those are comprised of, etc. And so that's why, a very similar system could be used on RNA or on proteins or on anything else, right?
So that leads me to the question, why RNA and why not proteins?
I think that the protein space is very interesting as well, in many ways. However, we specifically identified RNA as much more neglected from this standpoint. Again, I think that's a concept that comes back fairly often. The number of companies out there working on AI for proteins, such as designing antibodies or doing, AI drug discovery of small molecules to bind proteins. There's a fair number of them at this point. And while that's very exciting work, if you look at the number of companies doing AI for RNA structure, it's very limited. I mean, you might argue that to the best of our knowledge, we're the only one today. I don't think that's going to be the case for much longer, but it's a very interesting position to be in.
Okay, now let's talk for a minute about what specific types of RNA you're interested in, right? Because, people who've heard about RNA and know what it is, I think there's a misconception that the only role, right, is as a messenger, right? Carrying genetic information from the DNA into the nucleus to the ribosomes where it gets translated into proteins, right? This is
The
standard
central
dogma,
Yeah.
right? But there's messenger RNA and then there's non-coding RNA, right? And it turns out that there's many roles for non-coding RNA in biology. I mean, could you explain some of these and say how non-coding RNA fits into healthy cellular functioning and disease?
Yeah. Overall, there's these interesting stats out there that you can look at. And just before I dive into the specific mechanisms, but if you look at the entire, human genome, about 1.5 to 2% of it becomes translated to protein at some point or other, versus about 80% of the genome gets transcribed to RNA, essentially. So there's this vast space of RNA biology that's still actually relatively poorly understood. And it represents really this opportunity to find that novel, interesting biology. To speak about specific mechanisms, right? One very obvious one is you have the mRNA, right, which has this coding region in it, essentially, which is the part that codes for the proteins. But off the ends of these, there's these regions called the untranslated regions, or UTRs for short, right, that are, essentially are not translated. The name is what it does, essentially. And those pieces are oftentimes responsible for regulating various aspects of, how quickly the RNA is degraded, how much protein is expressed off of a given transcript, or even things like determining where the RNA gets localized within the cell. Right? And so overall, there's a number of these elements, these non-coding elements, even attached to existing, mRNAs that are responsible for regulating a lot of that function. Another example, right, is ribosomal RNA. Right? The ribosome itself, for translating from RNA to proteins, is itself primarily composed of RNA. Then you even have, as you were referring to, all this non-coding RNA that isn't even, part of mRNA, which, is this even larger, vaster world of, RNAs whose functions sort of are still quite poorly understood in many ways, but have a number of roles, that have been implicated in various cancers, for example.
So, other than trying to ask you for, or, what diseases might fit into this, right, from a non-coding RNA and where you could intervene, is why have these been, and I think these are called undruggable diseases up to now. Like, why has it been so hard historically to predict RNA structures and find molecules that bind to it?
Right. Yeah. So you mentioned this concept of undruggable, diseases. And in a lot of ways, people were, this oftentimes refers to people trying to drug the proteins implicated in a disease and being unable to do so. A classic example that people give of this is, the c-Myc protein, which is involved in, it's essentially overexpressed in 75% of human cancers. And we've been trying to drug this protein for a good 30, 40 years at this point, and essentially have encountered a great amount of difficulty in doing so. And that's essentially because the protein itself is mostly disordered, doesn't adopt a single structure, and the binding interface, when it does bind to other proteins and DNA, it's in fact a transcription factor, is very large and so hard to interrupt with a small molecule drug. And so the dream of RNA is you're instead, going one step earlier in this whole process, and instead targeting the RNA, the MYC transcripts that code for this protein, and decreasing the amount there instead, which is really the objective that you're going after, decreasing the amount of the c-Myc protein in this case. And so that sort of maybe at a very high level is the dream of RNA targeting drug discovery, or at least one of the mechanisms by which this happens. There's actually a few different ones. This dream, a number of companies realize this and have been going after this area for half a dozen years at this point at the very least. But they've encountered a significant amount of difficulties. In particular, they found it's hard to get a molecule that is selective to a specific RNA and doesn't bind many other RNAs, which has obvious toxicity effects, but as well, really getting molecules that are functional, that can bind those RNAs and actually, have the desired effect. And what's emerged is that to get over these problems, structure really emerges as a very key piece of the entire puzzle. Because if you've got a very unique 3D structure, first of all, that lets you very selectively target that structure and predict that. But then on top of that, right, if something is well structured like that, generally, nature doesn't waste effort. If it's built something that intricate, that likely is functional as well and is doing something interesting.
So now that we've sort of covered all the science, and I know it's not all the science, and some of the history background, let's come back to the technology at Atomic, right? Now, you've given it a new acronym. You're calling it PARSE, which is Platform for AI-Driven RNA Structural Exploration. So first, can you explain, I'm just thinking about this. PARSE is related to ours, or Aries, I should say. But does PARSE include the Aries, and in what ways does PARSE go beyond Aries?
Parse does include Aries, right? But in many ways, the updated name is actually a reflection of the progress that we've made on this core platform. Since the seed round that we raised, we've actually been laser focused on improving this core structure engine. And that includes both sort of improvements on the AI side of things, of new algorithmic advances that even have happened in the last couple of years, right? Even in the in-between times since the science paper came out, as well as advances on the data side of things, right? We've actually have in-house wet lab capabilities that have been generating data specifically tuned for the AI algorithm. And so part of the key of Parse is that it's no longer just a computational sort of algorithm. It's really an integrated wet lab computational technique with the two aspects really working in a tight iterative loop to really accomplish dramatically improved performance and speeds in terms of the predictions.
So what kind of data are you getting from the lab? I guess the two pieces together, and how does it improve? I'm assuming it's feeding back and it's improving the prediction algorithm.
Yeah. The data in the lab are these genomics-based assays primarily, which are designed for scale, maximizing information content, etc. And how we leverage those is that we're using them to, supply and train the AI algorithm. And then once you've trained the AI algorithms, you can actually then, further guide further rounds of experimentation in the other half of the loop and say, hey, we really should collect additional data on these kinds of RNAs, right? To really, maximally inform the AI and see where it's not confident, essentially, in this loop. On top of that, you can also use the experiments to validate some of the most key predictions, right? So if you're very confident you've got a structure somewhere, you can then go and validate that structure using more classical experimental methods. But overall, there's really this sort of key, integration of these two aspects of this loop, right? Of the experiments to train the AI then can guide further experiments as well as be validated by those experiments.
So here's the billion-dollar question, no pun intended, after raising that much money. But Aries was cool because it's so good at saying whether a predicted RNA structure was accurate. But now, how do you transform that into a drug discovery machine? I mean, how does Parse determine which parts of an RNA molecule are what, say, biologists call ligandable or targetable with a small molecule drug? I mean, can you walk us through maybe an example or what you're thinking?
The key overall is that while there are structured pieces of RNA, a large fraction of that RNA is also relatively unstructured and non-functional or non-ligandable either. And so really, I describe it as you're not only predicting those 3D structures, but you're also finding which parts of the transcriptome, right, the space of all human RNA, is structured and ligandable in the first place. And, this is really, you put it as a billion-dollar question. I actually think that's very, accurate in many ways, because there's, this dream of targeting RNA. But if you talk to, executives in the pharmaceutical industry, the difficulty is, say you want to target the MYC transcript, that cancer target I was just talking about before. You don't even know where to start. You don't know which part of the transcript to try and hit. And if you can hit it, if you can design a molecule that will be selective enough to hit it, or really accomplish the desired functions. And so really, you need a technique to find these structured regions across the transcriptome, find these unique ligandable regions. And if you look at classical experimental technique, wet lab techniques such as X-ray crystallography or cryo-electron microscopy, to name a couple, those, are very good at validating a single transcript or a single piece of RNA. If you're really pretty confident it's structured, that'll still take months or years if it's doable at all. But they're not really a tool for discovery. You can't use it to scan through entire transcripts to find those structured pieces. And so that's really where the AI plays a key role, is you're using the AI to scan through it and find those structured pieces in the first place. And then from there, you can validate it through experimental techniques if you really want to.
So now that brings the question of, okay, what's the business model for a startup like Atomic? I mean, your main asset is you have this proprietary machine learning model. Will you provide drug target leads to other companies as a service? Will you focus on discovering and developing your own drugs? And if it's the latter, is there a particular disease area you're targeting? I mean, I was reading the press coverage and I seen mentions of cancer and neurodegenerative, but I don't want to make any assumptions without you telling me what direction it's going in.
Of course. Really, how we're looking at this is through a partnering sort of focused model, right? We're revealing many structures across the transcriptome at this point, many more than you could ever possibly think of pursuing internally, right? If you have 100 structures, that's and each one of those could potentially become its own drug program. It's a little mind-boggling at some point. And so by finding the right pharmaceutical partners to work with, we can really partner with them and build out these programs in partnership, really. And, it's not like a service kind of model. It's really these co-discovery type of, drug discovery partnerships that we're looking at on that front. That being said, we also really want to focus on, validating these structures internally as well, and showing that you can use these structures to lead to, promising drug discovery programs. So we're also having our own internal efforts that we're pushing forward to really validate, the value of the structures we're discovering. And so overall, it's really this joint, this joint internal plus partnering kind of model that we're pursuing. And in terms of the specific areas that we're looking at, we're not disclosing at this time our specific targets, but there's a couple areas where RNA, targeting small molecules have shown a lot of promise. In particular, that's oncology, right? Neurodegeneration, neuromuscular diseases, rare diseases, and infectious diseases, actually, where there instead you're actually targeting viral genomes, RNA, essentially RNA genomes.
So, I mean, I know we have a broad base of listeners, and I just want to sort of maybe hit on a point to make sure there's no confusion. So I want to talk about how Atomic is different from, say, other biotech companies focused on RNA. I mean, I think a lot of listeners are probably familiar with, the kind of RNA targeting drug, namely antisense or RNA interference drugs developed by companies like Alnylam or Ionis. I mean, they make drugs where the business end usually consists of nucleotide sequences that are sort of complementary to the sequence known of some known RNA molecule. I mean, if you know the sequence of your target RNA molecule, you know in advance that your drug molecule will bind to it the same way that, say, two helices of DNA bind to each other, and then you can sort of interrupt its function. But you're talking about a totally different kind of binding that has nothing to do with the RNA's nucleotide sequence, right? I mean, can you say a little bit more about the difference? Why can't you use antisense or RNA interference techniques to target the kind of RNAs that you want to target at Atomic? Or, I don't know, maybe put differently, why isn't it enough to know the RNA strand's nucleotide sequence to know how to bind to it?
A lot of these ASO, siRNA, RNAi kind of technologies are, we refer to them as RNA-based medicines, right? The therapeutic is an RNA itself, which is, somewhat overlapping, but a distinct concept from, RNA targeting medicines, where you're, really designing something to target those. In our case, we're looking at small molecule RNA targeting specifically. Right? Designing small molecules to go after, RNA. And, these RNA-based medicines, such as ASOs from Ionis. They've done amazing things, but a major challenge with them is essentially delivery. Getting those to the right place in your body is quite difficult. Essentially, you can target the liver, right? So everything ends up in your liver. You can target your the eye, because that's a relatively close system. But for for targets outside of that, it actually becomes a major challenge. To give a quick example here, spinal muscular atrophy, right, or SMA, right, had an approved ASO from Ionis, Spinraza in that case, but the delivery vector was essentially, you essentially needed, this surgery every month to deliver the ASO payload, essentially. And then now instead they've approved a small molecule to also, target, very similarly SMA. And in that case, right, and now it's just a pill in a bottle that you just take orally. And it's just the delivery system is just much simpler in some ways because we understand how to deliver small molecules in a way that these RNA-based therapeutics are still, it's very much an open research question. The difficulty, though, as you alluded to, is, for these ASOs, you find the complementary sequence, you can target it that way, versus to get a small molecule targeting a given piece of RNA, there's no such thing as a complementary sequence anymore, right? There's just much larger space of chemistry that you need to explore. And so to target it productively, you really need to find these unique binding sites, right? These binding pockets, really, is how we refer to them. And that involves you need to model the structures, really, at the end of the day.
Interesting how we always seem to come back to structure being something that gives us a lot of insight about how to attack
it's how
this
I view
thing.
the world at the very least, maybe
Well, no, but I
biased
mean,
perspective.
you look at, AlphaFold and you think about, there's, all these areas where we're understanding how the thing is shaped, and it really gives us insight about, okay, how am I going to attack this beast sort of idea, right? But so what's next for Atomic? Do you already have a pipeline of drug candidates? I don't know, what sort of milestones are you thinking about for the company? And I guess if I said, hey, if we were talking a year from now, what do you hope would be different?
We are really at this point, at a fairly early stage in the drug discovery process. We're just right now, sort of, developing our emerging pipeline of programs. We've really taken this data-driven approach of scanning across the entire, disease-relevant transcriptome to find which RNA targets are ligandable. Actually, a very important point is that, if there is a transcript that is of interest and we don't find any structures in there, that's also a very important piece of information, because that tells you should not waste your effort trying to go after that one in particular. It could save you a lot of pain and suffering down the line. So in terms of milestones, really what we want to do over the next coming years is start pushing forward these internal programs and really demonstrate the validity of these structures that we're finding and connecting those to the downstream sort of functional impact that we're very much after. On the platform side of things, we're also looking to continue and expand our capabilities. There's many related sort of problems beyond just predicting a single structure that all have to do with machine learning on atoms and RNA generally. And so expanding those capabilities is also a very key direction. You alluded to one earlier of, it's not just about predicting a structure, but finding where something is structured across a transcript, which is, a related but somewhat different problem. Another one is that RNA is a very flexible molecule. How do you model, if it's adopting multiple shapes? And that's really linked to a lot of this exciting new sort of generative modeling kind of technology that's been emerging on the AI side of things over the last couple of years. So there's a sort of whole wealth of sort of problems to attack on machine learning on atoms in this RNA space that we're really excited about. And that's perhaps, linked to the name Atomic AI at the end of the day.
Well, it'll be interesting to catch up, a year from now and find out where you guys are and what you're doing. I mean, I think, I wouldn't be doing this if I didn't think it wasn't super exciting, and going to be very impactful to, how do we treat certain diseases and, help patients, get better. So,
Yeah.
I can only wish you incredible luck, and it's been great having you on the show.
Yeah. Thank you so much for having me. It's really been a pleasure to be here.
Excellent.